Okay students, now we will do the activity to test for the presence of starch in biomolecules. As food samples, I have 3 samples here. A piece of string hopper made from flour, a piece of potato and flour for starch. Now you all know we need to use one particular reagent, that is a solution of iodine, which is reddish brown in color. The procedure is to add few drops of iodine to the food sample and observe. Before I start the activity, I will also add a small sample of flour onto the tile. Here we are using a white tile, so you can see the color changes also clearly. Now I draw a drop of iodine and add it to the string hopper. Another drop of the iodine solution to the piece of potato and another drop to the flour. You can see the color change, the appearance of purplish blue color or dark purple color in the piece of string hopper as well as flour, but the color is not very visible in the piece of potato. So I will add another few drops of iodine. Here you can see slightly the purple color appearing on the piece of potato. If you look at the center of the potato, you can see the color change. So students, you can observe all the 3 samples. Observe clearly the color that has appeared, a very dark purple color. This confirms the presence of starch in the food sample. So when you use iodine solution, the reddish brown color of the iodine solution should become a purple color. That will confirm the presence of starch. To show the difference between the normal color of iodine as well as the color change with starch, I will put a few drops of iodine on the tile. You can see students, a light reddish brown color at the same time. With the 3 samples, you can observe the purple color. So you can see the color change very clearly. Okay students, now we will do the activity to test the presence of glucose in food substances. For that, we will need glucose. To dissolve glucose, water. Then the reagent that we are going to use is Benedict solution. You can see it has a nice blue color, and that will give a color change with the glucose solution. So to prepare the glucose solution, I will need a boiling tube. We will be using a boiling tube because we have to heat the solution. First, I will prepare a solution of glucose in a boiling tube. We will need a boiling tube because we have to heat the mixture. So to that, I will add certain amount of glucose into the boiling tube. And add water. As you know, since glucose is a monosaccharide, it can dissolve very easily in water. And we make sure that it dissolves well. Now that the glucose is dissolved and we have the glucose solution, to this I will add a little amount of Benedict solution. When we add a small amount of Benedict solution, you can see it has a nice blue color. But the observation of the activity will be after heating the solution. We can directly heat this solution in the flame, but that will give rise to a very fast color change and you will not be able to observe it clearly. In order to avoid that, we use a water bath. A water bath is a beaker containing water and we heat the water. So gradually the heat from water will be transferred to the solution mixture. To place the boiling tube inside the beaker so that it will become hot gradually. Now you can observe the color change clearly. You can see at the bottom of the boiling tube there is a light green color appearing. So the mixture will completely become green in color first. After green, you can see the appearance of yellow color. I will mix the solution slightly so that the color is uniformly distributed. The yellow will now turn to a brick red color and finally there will be a brick red precipitate appearing. You all can see the solution has turned from yellow, it has turned almost to a reddish color. You can see, students, the appearance of a brick red color. Now you can see the color change clearly. The solution has become brick red in color and the precipitate will deposit at the bottom of the solution. So students, you have observed the color change clearly. The blue color of the copper sulfate became green, then it became yellow green, finally it became a brick red color precipitate. So this color change confirms the presence of glucose in food samples. Okay students, now we will do the activity to test the presence of sucrose in food samples. For that, I will be using sugar, the normal sugar from the household kitchen, that is sucrose. And the reagent that we are going to use is Benedict solution. In addition to that, I explained to you all, we will be needing dilute sulfuric acid. First, I will prepare a solution of sucrose by taking a small amount of sugar into the boiling tube and adding little amount of water. As you know, students, sugar can dissolve very well in water. So if I mix it nicely, you will get a proper solution. Now that the sugar has dissolved well and we have the sucrose solution ready. To this, I will have to add a small amount of the Benedict's solution. We have used it before also. You can see the color clearly. It is a blue color. solution. So I add a small amount of Benedict solution. When it is mixed, you can see it gives a nice blue color. Now I used a boiling tube to make the sucrose solution because we have to heat it. For heating, we use a water bath. As you know already, water bath is needed because we need to observe the color change slowly and clearly. If we heat it directly, then the color change will be too fast. So for water bath, we use a beaker containing water. I have already got it ready, and you can see the burner as well as the tripod and the wire mesh. I will place the mixture into the beaker. You can observe the color change. We will have to wait for a little time. Students, you can recollect the color change that has to be observed here. As you can see, there is no change taking place. Still, you can see the blue color of the Benedict solution only. As you can see, students, even if we keep on heating this for a long time, there will be no color change because sucrose is a disaccharide. Then to obtain the color change, we will have to do the second part of the activity. So for that, I will again need a freshly prepared sucrose solution. I will remove this from the water bath and I can just place it in the test tube holder. We will just leave it aside. Now I am going to prepare another solution of sucrose. The sugar has to be taken into the boiling tube again. And when I add water, we get a freshly prepared solution of sucrose. Now that the sugar has completely dissolved in water, we will add a few drops of dilute sulfuric acid. Here you can see this is dilute sulfuric acid, and I am going to add a few drops of it to the sucrose solution. And then I will be adding a small amount of Benedict's solution. Now we will heat the mixture using the water bath. Already I have prepared the water bath. We used it for the first part of the activity. Now observe closely. You will see the color change clearly. Try to recollect the color change, students. Now you can see the solution slightly turning from blue to green color. First it was a blue colored solution. Now it has become a green color solution. The color change will continue. You can see the solution changing color from light green to a yellow-green color. Now you all can see the yellow-green color becoming a little more darker. Now students, you can see the color changing from yellow to orange. If I mix the solution properly, the reaction will take place more uniformly. And now you can see the appearance of a brick red precipitate at the bottom. Although the solution is orangish in color, you can see a deposit at the bottom of the boiling tube. So this is the color change that you have to obtain. If there was glucose in the solution, the Benedict solution will change color from blue, then to green, then from there yellow-green, orange, and finally, like you see in this mixture, a brick red precipitate. So from this activity, when we added sulfuric acid, then Benedict solution and heated the mixture using a water bath, we got the color change. So from these students, you can understand what has happened to the sucrose in the sugar. With the addition of sulfuric acid, sucrose has been hydrolyzed to glucose and fructose, and the glucose gave the color change with Benedict solution. So you have seen the observation clearly and you have understood why we did this activity in two parts. First there was no color change, then we saw the color change very clearly. There is one more step that you can add to this activity. In case you have added an excess amount of dilute sulfuric acid before in the previous step, then we can add sodium bicarbonate to neutralize that acid, and thereafter we can add the Benedict solution and continue the activity. So that is an additional step that you might have to remember. Okay students, this confirms the presence of sucrose in sugar. Okay students, now the activity to test for the presence of protein in food samples. As you know, there are different types of food that contain protein. Dal is one of those substances. You can also use egg white for this activity, but for now I'll be using dal. Since dal is a solid substance, we will have to get an extract of the dal. For that, I will need a mortar and a pestle. First, I am going to transfer a small amount of dal into the mortar and pestle. And I will be adding a little amount of water to it. I will have to grind it well and get the extract. To make it easier for grinding, you can also soak the dal in water for about 2 to 3 hours. Then it will be more softer and it will be easier to grind. So now I am going to grind it and get the extract. You will have to keep on grinding it for quite some time so that the substance from dal gets extracted into the solution. Now I have ground the dal into a nice solution and extracted the substance from it. I will decant this solution. Decant means just transfer the solution into a beaker. Now I will not be needing the mortar and pestle. I will put it aside. Now we are going to do the test to identify the proteins present in the dal solution. For that, I'll be needing two reagents. You all know the reagents. This is copper sulfate. Copper sulfate is blue in color. It is a nice blue color solution. And sodium hydroxide. First, we will need the sodium hydroxide solution. I'll be using a test tube, taking a small amount of the dal extract and into another test tube, you will have to take either equal amount of sodium hydroxide or slightly excess amount of sodium hydroxide. So, I'll just use the two test tubes to compare. Here you can see I have taken slightly more sodium hydroxide than the dal extract. Now, I will transfer the sodium hydroxide into the dal extract. You can still see the color of the dal extract. Now, to this mixture, I will be adding a few drops of the blue color copper sulfate solution. Now, observe clearly what will happen. You all can see a nice color change, the appearance of a purple color. So, with sodium hydroxide and copper sulfate, if a purple color appears, that means that sample contains protein. And you know, students, what is the name given for this test? Yes, this test is called the Biuret test. Now, we will start the activity by taking 2 milliliters of the starch solution. So, before taking the solution, I will mix it well. Since this is not a quantitative activity, I will take a small amount of starch, about 2 milliliters. To this, we will be adding 2 milliliters of the germinating green gram extract, which contains amylase. So, now I have equal volumes of starch solution and the amylase extract. If you all are doing it in a group, one person can mix it. Simultaneously, another student can start the stopwatch, because we will need to time this activity and do the testing every 2 minutes for 20 minutes. So, now I will mix solutions and immediately start the stopwatch. So, now the reaction will take place between starch and amylase. While we wait for the first 2 minutes to be over, I will show you all the color change that will take place between starch and iodine. Iodine is the reagent that we are going to use for the testing. So, this is a starch solution that I used. I take a drop of the starch solution so that you can again look at the color change. To this, I will add a drop of iodine. Now, you can see it is a very dark blue color, almost blackish blue. This is only for our reference purpose. Now, we will get ready to take the first drop from the mixture and test for the presence of starch. So, I have the starch amylase mixture. Now, 2 minutes is up. I draw a drop from it. To this, I will add a drop of iodine. another drop to see the color change clearly. Here also you can see the formation of dark blue color, but comparatively to the starch solution, this is somewhat lighter. A slight difference can be observed. Again, we will have to wait for another 2 minutes to observe the next color change. Now, end of 4 minutes. So, we will take another drop from the reacting mixture. I will place it here. And to this again, we will add iodine. Here also you can see the formation of the dark blue color, but when you compare the 3 samples, the first one pure starch is very dark, after 2 minutes is a little lighter, and you can see after 4 minutes there is a slight difference. We will have to wait for a long time before we see a distinct difference. So until then, we will have to draw samples from the mixture every 2 minutes and test it with iodine. Now we have waited for 20 minutes. Again, we'll test with another drop of the mixture. This is again a mixture of starch and amylase that has been reacting for 20 minutes. To that, we'll add a drop of iodine. You can clearly see that it gives a reddish brown color. So that means from this observation you can conclude that there is no starch in this reaction mixture. So as you know, amylase is an enzyme that acts on starch and hydrolyzes it to maltose. All the starch that was present in the reaction mixture has been now converted to maltose. So that is the reason we do not get a color change. Only the color of iodine is visible. So with this observation, students, we can clearly understand the activity of amylase on starch. Okay, students, now we are going to test for the presence of lipid in biomolecules. So as constituents of biomolecules, we take food samples for our test. And as you know, lipid, there are 2 forms, the fats and oils. So I am going to use 2 samples here. One is butter, an example of fat, and coconut oil, which is an example for oils. And the reagent that we are going to use today is Sudan 3. I have already taken a small amount of Sudan 3 into a test tube. You can clearly observe the color of Sudan 3. It is a red color solution. First, a small amount of oil is taken into a test tube. To that, we have to add few drops of Sudan 3. Here you can see the Sudan 3 moving down along the oil, and when we mix it well, you can see the appearance of red fat globules. Here, students, you can observe clearly the appearance of red fat globules. I will add one more drop. Here you can clearly observe the red fat globules. So this is the observation for the presence of lipid in food samples. When you add Sudan 3 reagent, there should be appearance of red fat globules as you see here. The globules are going down from the top. So that is a very simple activity to test for the presence of lipid in oil or oil as a lipid substance. Now we will see the test for solid lipids, that is fats. You cannot use the same reagent because this is in the solid state. So what we will be doing is we will use a white paper. Any white paper is okay, and we take a small sample of butter onto this white paper. When you fold the white paper and rub both papers together, you will see this translucent patch. Here you can see a translucent patch. This is the translucent patch. This appearance of the translucent patch confirms that the substance we used is a fat. In other words, it is a type of lipid. Okay students, now we are going to do an activity to test the presence of water in food samples. For this, you can use any type of leafy vegetable or fish, meat, or even any type of leaf, because all living organisms contain water in their body. Today, I am going to use some leaves from a garden plant. So, these are the leaves, and we should make sure that the sample, if it is a leaf, that we don't wash it prior to the activity. If we wash it, we wouldn't know whether the vapor that is coming out is from the water that is there on the surface or from the sample. So, we will have to make sure that we use a dry sample. And then, we will have to heat this for a long time. So, I am going to use a crucible with a lid. A watch glass to make the vapor to condense on it, and we have anhydrous copper sulfate which has to be white in color. Also we have the pink color cobalt chloride solution, but we will have to dip a piece of the filter paper in cobalt chloride, and then I will heat it so that it becomes anhydrous cobalt chloride which is blue in color. So, the reagents here are anhydrous copper sulfate and anhydrous cobalt chloride that will be made from this. Now, to start the activity, I will take the crucible. Then, into the crucible, I will put few pieces of the leaf. If we break it into pieces, it will become hot quickly. Then what we do is we place it on the tripod on top of the wire mesh, and I will close it with the lid. After it gets heated up for some time, we will remove the lid and cover it with the watch glass so that the vapor that is coming out will condense on this watch glass. It has been heating for some time. I will remove the lid of the crucible using a pair of tongs and place this watch glass over it so that you can see the vapor coming out and condensing on the watch glass. We will collect that vapor. Now you can observe, students, that there is a liquid that is vaporizing coming out of the sample and condensing on the watch glass. I will remove the watch glass now. you can clearly observe the droplets of liquid, and to that I will put copper sulfate. You can see the color changing. The light color of the copper sulfate, that is anhydrous, is turning to dark blue color. At the same time, I will dip a small piece of paper. When I hold it to the flame, you can see the pink colored cobalt chloride. You can compare and see becomes blue color. If I touch it on the drops of liquid, again you can see it changes color. So here you all can see, students, the copper sulfate has become blue in color. This blue part is the anhydrous cobalt chloride. Wherever there was water, it has become pink in color. So these two observations confirm that the vapor that came out and condensed on the watch glass is water. So with this observation, we can conclude that there was water present in the leaf sample. That means there is water in food samples as well as living organisms. Okay students, now we will do the activity to test for the presence of carbon in food samples. For this again, you can use any solid food sample, leafy vegetables, piece of fish, meat, anything can be used. Today I am going to use these leaves. I will take a few of these leaves, and you all know for high heating we will be using a crucible with a lid. I will remove the lid of the crucible and break these leaves into pieces and put it into the crucible. Place it on the wire mesh of the Bunsen burner and place the lid. While it is getting heated up, we will have to remove the lid on and off so that the water that is vaporizing from the leaves will be removed. I will remove the lid on and off. You can see the water vapor coming out. Again close the lid. Leave it for some time. Again I have to remove the lid and allow the water vapor to escape from the crucible. Like this, we have to keep on heating it until there is a change in the substance. Again, if I remove the lid, you can see that the leaves are turning in color. They are almost becoming brown in color, and when we keep on heating it, they will become more and more dry, and finally we will end up with our required end product. I will close it again and let it to heat. Now again I will remove the lid. You all can see, students, that the sample has become black in color. We will leave it for few more seconds. Yes, you can observe nicely there is a black color residue that has been formed inside the crucible. Now that all the water has become vapor and has been removed from the sample, there is only a black color residue remaining. Then I will have to remove the crucible from the burner. You will have to leave it for some time because it will be very hot, and after that, once it cools down, we can take out a small piece of the black color residue, and then I will try to draw lines on this white filter paper. I think by now the sample must have cooled down a bit. I will take out the most blackish color parts, place it on the paper. You have to make sure it is not hot. Then I will draw some lines with this. Here you can see clearly that there are black colored lines being drawn on the filter paper. By now you would know what it is. You have seen the observation. Black color is carbon. Carbon graphite, what we normally get from burning firewood, is also black in color. When you burn firewood, we end up with coal or charcoal. So this charcoal, if you draw it on a piece of paper, you will get similar black colored lines. So from this observation, we can confirm that the plant leaves have been converted to charcoal. Charcoal means they are in the form of carbon. So that confirms there was carbon present in these plant leaves. Okay students, now we are going to do the activity to test the presence of nitrogen in food samples or the presence of nitrogen as a constituent in biomolecules. We have already done this test with proteins. You know the name of this activity. What is it? Yes, it is biuret test. So I will be needing a sample that has protein so that we can test for the presence of nitrogen, because nitrogen is a constituent of protein. The reagents, the two reagents, sodium hydroxide solution and the blue color copper sulfate solution. I have already taken the egg white into a small beaker. Now I will transfer the egg white into a test tube. Okay. Now I will take another test tube and into it the same volume of sodium hydroxide or slightly excess volume of sodium hydroxide has to be taken. Here you can see students, it's almost the same volume, if at all a slightly excess amount of sodium hydroxide. Now I will transfer the sodium hydroxide into the egg white. To this mixture, we will add a few drops of copper sulfate. Now you can see the observation clearly. Can you all identify the color change, the observation? Yes, the appearance of purple color. This confirms the presence of protein in the sample. And you know that protein contains nitrogen as a constituent element. So therefore, we can conclude that this sample contains nitrogen in it.